Yeah, now the voice is also clear, and okay, that point is also started. Okay. Just related to practical sessions. Okay, let me just take you there then. Yeah. So I'm going to discuss. Actually, discuss particularly. So, though I have going to take this point, there is a practical sessions in afternoon time or in a noon time nearby. So we have going to have the total 9 experimentations are there. First is like a what? It is a preparation of solid and liquid medium. What is the basic things? Earlier I have stressed as the difference between solid and liquid, right? So it is the kind of a things when we are going to use the liquid medium, when we are going to use the solid medium. That's the basic objective one should clear. We are going to learn about them. That is like a related in second units we are going to learn. But in the first unit, it is related to nutrition of the bacteria. So we are going to I am going to discuss briefly what kind of the mediums are available. Like for our nourishment, we are having the like so many kind of food availability is there. We may sometimes classify in a non-veg and non-veg. Okay, if it is there in a veg also, so many varieties are there. In non-veg also, so many varieties are available. So like that only. For all the organisms, so many kind of like a phototroph, autotrophs, or the organotrophs or lithotrophs, or we are going to work as a chemotrophs or the chemolithotrophs. So many things, the varieties are available for all. Some are kind of a nutritive materials are available very easily, readily, and it is same for all. Okay, but some are very special. They don't like it, or they are not going to grow. Or some are the slow, yeah. Some are slow grower. Some are very fast grower. They will. It is like that. Those who are eating very fast. Just started, lunchbox is opened. Within 5 or 10 minutes, everything is get set up. Okay, some are taking time. Half an hour, hours. It is not completed. It may happen. Okay. Otherwise, they are starting to look for the another tiffins, and they will eat. They will keep their own for a while. So many things are there. Same like that in bacterial conditions is also there. Bacteria, some are the slow runner. They will eat their own food or they prepare their food and they will start to utilize. Then they are going for the growth. So the two terms are there: growth and development. And another part is related to reproduction. So it is like that. All the cells, like in mitosis, meiosis, we are learning, right? So here the bacteria majorly going into process like a cell or bacterial fissions. They are going to divide into like a mitosis. Mitosis, we are knowing there is a nuclear fissions also coming, right? But here they are not having the nucleus. So bacteria are going to divide. Binary fission is a very well-known phenomena. Maybe you are knowing or not knowing. So they just following it. So all the bacterial cell not following this, not going to divide in the same time period. Like E. coli, we may know like a 20 minutes. That from 1 to 2, their divisions and the two cells are going to form maybe in 20 minutes. If it is there, another bacteria is there, or the another from the protozoan, single cell amoeba. They may take a time about, let's just take an example, 8 hours. So what they are doing for the 8 hours? They are just resting or they are preparing themselves to get divided. So what is their motto in actual? Like a viruses, currently we are knowing like a corona, right? Corona has spread throughout the world. How it is spreaded, we are knowing the phenomena. But what was their motto? Just to divide or to develop themselves? Or they do have their some intellectuality? What to do, they are not knowing. But they are just doing, they want to survive. All the species on the earth just looking for their survival. So what they are doing for the survival, the time limitations for them, it is certain period. From the mother to daughter cells is going to happen. Okay. Similar to it is in higher animalia or eukaryotes we are looking, even in a human being. All are looking for the survival. If the some people has got died because of some certain reason, that space is going to get occupied by the anothers. Okay, so all the living species are looking for the survival only. The survival is possible what? That is, they are going to they propagate themselves what they are to another. So they are having their child. Offsprings are there. Okay, so that is the process is going on. So like the bacteria, no any kind of a species having any particular kind of intellectuality in that conditions that what will be there next. If it is a propagation is goes on. Okay, what's going to happen? But this is at the second and tertiary quaternary level happens. There is a like we know like a Darwinism, Darwin's principle, survival is the fittest. So overpopulations, overgrowth, there is some scarcity related to some nutritive material, habitat, or the space is going to happen again and again. So there is some are maybe a possibility. There is an internal or external forces are making them to get in some difficult situations. So those who are able to manage in that some difficult situations, they will survive. They will propagate and goes on. Those who are not, they are get extinct. So we know that right now we are going to have the some books, red, green, purple. So many books are coming that are extinct. Those who are species to be get extincted, those who are in a conditions to be get extincted. There are the WHO and the UN is maintaining such things. For all the categories, they are happening. So this sort of the things is there. So microbes are or the other organism not looking for what going to be next. If the whole world, if it is a surrounding, is get filled with the microbes only. If it is so, okay. If it is there, we are going in another planets. Like in currently, people are going in the moon and others. They may get the, some bacterial suspension and they are start to grow. By how? By chance, if it is just split down, or happens something, and the whole entire planet it is full of some microbes, one kind of species. Is it they able to survive then? There is only one species on a entire planet. Suppose we went through some nearby planets. Okay, it's XYZ, and they want to whether they able to survive one species? No, because they are having the whole space. Everything is there for them. What sort of two things are not what? Food chain. Okay, what they will eat. If suppose it is just they are eating the soil, they just want to nourishment. Okay, just think like they are in condition of chemolithotrophs. Now they can. I'm giving the one step ahead. Chemolithotrophs. You are maybe now knowing. We are going to understand their process and materials. That means they are using the chemical energy and they are nourishing by themselves. Can chemolithotrophs can survive single species on the Earth? On the new planet? They can do new planet. If every habitat, everything is changed, then they survive or not like that. Okay, one good point. Okay, he's telling about it. That is also one important point to be get understood also. That yes. So, how the species can survive without water? Okay. Anybody else want to add something? It is like a brainstorming. Pardon again. Okay, no predator is there. That is the luxurious, no? That they will not be controlled. They will okay, that means there are competitions within. Okay, that means you want to say. One is telling the no water availability. Okay, another is telling that internal competitions, and third is telling about that what could be the, I'm going to define in another word, physiological conditions. That means soil is there, Okay, but if it is there in a materialic form, it is a hard rocks or in a compartmentalized. Okay, very fine soil is there. There it is really difficult. Maybe that is like a pH temperature. Temperature is very high. Do your work. You won't. It is like that. There is some condition as well. If the light is not there. Okay, and the temperature is maybe it is like in Canada and somewhere else. You are knowing that it is a hot bubble is going on around 45 to 49. For us also it is really difficult. We can up to the 40. Okay, with up to the 40-degree temperature, we can manage. Not able to do a particular work, we can manage. But if it is going higher, our efficiencies get down. Those who are in the lower temperature is their temperament is there, and their temperature is working like in a western part we are going. It's near about 10 to 20. For 20 is very high. If they are going in a higher temperature and higher, won't be able to survive. So like that only, if they are going in another planets. Okay, so that was related to these 5 different our experimentations are there related to that only. That's one organism I have taken. Suppose it's E. coli. We are going to work generally it's E. coli. Okay, we are going to work on E. coli. If I'm going to change their temperature, fifth experiment is effect of temperature on growth. I will take it. I'm going to put in refrigerator. Class I will prepare. I will going to put in the refrigerator. One maybe in incubators. One in a lying in a room. Okay, maybe in a laboratory we are having the drawers. We going to keep there, and one maybe in a different temperature. Incubators are there. Suppose 50 degrees centigrade we have kept. So it is like a different planet they came. They don't know, right? Artificially we have created the environment outer side. For them it is a new planet. Now your logic will start, because they are able to generally 35 to 37 degrees centigrade temperature is better for them. Now they have moved into new planets. Okay, what will be the things? Growth will be get either retarded or get slow. Okay, either one is going to happen or it may increase. How going to increase? We don't know. So growth rate, the terminology we are going to learn in a that way. Growth rate. Rate means with the time. Whenever it is the rate terminology is coming in connections with the time. Within the time, what going to happen? Maybe we can take the readings. Here we are going to take the readings using the spectrophotometer. Okay. I think you people are knowing the spectrophotometers. Theoretically, at least. Okay, theoretically you are knowing the spectrophotometer. B.S. law are there. Okay, we are not going to technicality of it. We are going to use their applications. Okay, applications point of view, we are going to talk about it. What could be the in spectrophotometer we are going to use. So what we can do. There we are going to have one experimentation that is called the fourth experimentation, study of the growth of E. coli by using the turbidometric analysis. The turbidometric method analysis, so many terms can be possible. Okay, turbidometrics, turbidity. What is the turbidity? Like the solutions is there with you. Huh? Pardon again? Impurities. Okay, that can be also possible. Yes, changing of colors. So that means turbidity, we are going to learn in a way that like any solution is given to you. Okay, any solution is there with you. When you are going to keep in the spectrophotometer, it's going to measure there two terminology we may learn: absorbance and transmittance. Every elements or the chemicals or the molecules having the capacity to absorb certain light. Remaining is transmitted. So we cannot measure the absorbance, we are going to measure the transmittance. So transmitted light we are measuring. So when you are going to keep the NEA control, like in a spectrophotometer, two things are there. Two cuvettes. Maybe you are knowing the cuvettes. Cuvettes is like a small, maybe a round or the square bottom. One tubes are there. Small tubes are there. Yeah, spectrophotometer, there another is a colorimeter. Basic model, it is called the colorimeter. Another is the spectrophotometer. Colorimeter is working in a Vibrio. That is a visible spectrum. When we are looking at the spectrum, it is also coming the component of the UV light. So whole a spectrum you can cover up. Okay. So what's going to happen? The colorimeters, you may have the small tubes also. So when the company, the colorimeters are there, if you are using the one to another model, some kind of a size of the that covers may change. So what is the basic purpose over there? If you are going to use the water, simple distilled water, if you are going to cap, that will be a 0-0 transmittance, because there is a nothing over there to absorb anything. If some a reading is showing, what we are going to use, that nullify. Nullify that means we are going to make them 0-0. So water is our control. And then you are going to keep any another solutions. So it's going to give the some transmittance. That reading we are going to take is a 0 hour. At the 0 hours, if you have taken the nutrients broth, and broth, we are going to learn into it that what are the nutrients broth are there. It is like a nourishment material. Liquid material is there for the bacteria. Let they grow. Okay. So at the point, we are going to take the nutrient broth as some reading. 0 hour. Now you have inoculated certain bacteria. Population is there. And you have kept one at a refrigerator, another at the incubator, and some in the room temperature and somewhere else. So for them, new planet is there. After 1 hour, what you have started to do, you may take that flask from the incubator and start to measure their reading. Colorimeter is there. If you are going to go there and you are going to again take the readings, you will surprise the reading has changed. You are going to surprise. The reason is what? That means after 1 hour, bacterial population has started to grow at the 37 degrees centigrade, because for them it is the regular habitats. But for other place it will be changed. Okay, in a refrigerator you have kept the reading may be near to 0-0. That means bacteria were not grown. Another incubator you may have kept at 28 degrees centigrade or the room temperature. There the compared to 37 may have some drop down. Reading change. That means some numbers will start to change. So that reading, if you are going to take for about 24 hours, we are not going to. But suppose 1 hour you are coming, for the 20 hours you are going to check, you will find that up to the certain time period, there is a continuous growth will be there. Growth of the microbe will start to grow. That is exponential phase is going to happen. So bacteria can survive normally into their normal habitats, but at not others. So we are going to learn their effect. That first point is the temperature. At which temperature they will grow very well. Another next point is there related to our osmotic pressure. I think, okay, let's first complete temperature only. Is it clear? Effect of the temperature on the growth of any bacteria. That means you are going to keep in the refrigerator, turbidity or the reading in the spectrophotometer will just low down. For 24 hours, if you are going to take like a flask you have kept, you have to take the 1 or 2 ml out of it from that flask, and you have to measure their turbidity. Maybe for the 24 hours, after a 12 or the 14 or 16 hours, slight change may happen. In the refrigerator also, when you are going to take the readings, it may happen that slight variations may come. That means bacteria start to grow at the lower temperature even. That is why we are knowing that in refrigerators, maybe after you are coming after the 1 month, you are going in the holidays and coming back, when you open your fridge, you may found the stale food. Right? Food has also get rotten up within the fridge. You are get surprised. What happened? Fridge is not working? Or any problem is there? No. So bacteria are try to survive into lower temperature also. Okay, so they have started to grow up. So the temperature has done some effect on their growth. Growth is either halted or retarded. Okay, so in retarded means there is a total zero. No, halted means in a pause mode. So for a while it is in pause, but then after they start to grow, they have start to manage within certain restricted conditions. If you have kept yet one another flask, and that you are going to keep in freeze. Then the refrigerator, two components are there, right? At the lower bottom, or the now the very patterns are also changing in freeze. That means in a freezer, if you are going to keep, what going to happen? What do you think that E. coli will able to grow after 24 hours also? Huh? Please louder somewhat. Growth will decrease. Okay. In a refrigerator, it was there. If I'm going to keep my flask in the freezer, there be a growth of the microbes will be there or not. It will be decreased, retarded, or halted. Three options I'm giving. Freezer, ice, where we are keeping the ice, right? Retarded or halted.
Should be halted.
Mm-hmm. So, okay, I'm repeating the question. I have prepared one flask, 250 ml of the flask is there. I think you are knowing the amount, right? 250 ml flask I have taken. I have poured the 150 ml nutrient broth over there. I have inoculated with the bacterial suspension, and then I have kept this flask in a freezer where I'm keeping the ice. Now, after 24 hours or the 48 hours, the growth of bacteria will be there or not? First question, yes or no? Okay, wait. Yes or no? Or you can say if it is a yes, okay, then it will
No,
be
sir.
okay. Some are speaking.
It will be no.
Okay, no. Okay. Some people are telling that the growth will be no. Okay. If it is there, if it is coming as a yes, it will be in a retarded form or in a halted form. Now the second question. Retard.
They are supported in the temperature of the freezer, then it will grow. But if they are the types that are which need higher temperature, then they will not grow.
Okay.
But on the other hand, if it is supported in the higher temperature, then all it will get retarded, all it will get grow, and then it will stop.
So kind of the bacteria, it's okay, fine. It's dependable kind of bacteria, but I am giving the example of E. coli only. I am talking about E. coli only. Okay, E. coli. Okay, that means you are telling that they want to grow. Your answer is that there will be no growth. Okay, if no growth, then also two questions again. It is considered as a halted or retarded. Means what? Retarded means total zero. Now they lost the capacity. Now let me open that part. Retarded, that means they have lost total capacity to grow next. And halted means when they will going to get their normal temperature, they will grow.
They are halted. Halted, sir.
Halt means post. Okay, wait. When we are looking for same terminology in microbiology, biochemistry, whenever we are going, various terminologies are having their another explanations related to this subject to context. You are right, retarded. Right. That is, you are talking in the English literature. Right. Same thing. We are going to in genetics also. I think you may have some genetics offspring we are telling. Childs. Okay. Then the progeny. So many terminologies are there, but they have word to context, or you can say the subject context. There is some explanation is changing. That means retarded, halted, or otherwise you can say the lost their capacity to grow. Okay, so they will be halted or they have lost their capacity. So first category possible, that means they have halted. 0 degree temperature or the lower temperature. I have kept one of my flask in the freezer. Media itself gets freezed. Just one simple. Okay. It should be in either in liquid or solid medium. Suppose bacteria can grow on the solid medium also. Okay, but the one things they have get freeze down, so they have get solidified, but they have maybe they lost may not capacity. That means you can say fully lost the capacity. You may say that no, they are able to grow. Now the last option is remain with us. That means in halted condition. Bacteria, it is in halted conditions right now. They are not able to grow at the lower temperature in freezer. But the same flask, if you are going to take out, keep in the incubator, they will grow. So what you have done, this is called the physiological parameters. Our whole paper is asking about microbial physiology. So what are the physiological parameters outside? This all are. One is the temperature, osmotic pressures, following the heavy metals, chemicals, and antibiotics. So many lists is there. Just for the experimental purpose, we are going to have the some limited part only. But then theoretically, we can think, because to perform them, it will be take more time. So at least 4 or 5 variants are there into same experimentations that we will do. Okay, so just temperature, maybe you have understood now, right? For them, it is a new planet. Okay, for them, it is a new planet. E. coli have gone into refrigerator. Oh my God, it's fully chilling. So they will wait. When the time will come, I will show my things. Okay, that means they in altered conditions. When the flask is taken out, they can grow, right? But if it is kept at 28 degrees centigrade, then physiologically 37 degrees is better for them. But flask you have kept at a 28 degrees centigrade. What's going to happen? Yes, growth rate will decrease. That means they will able to grow, but it will take time. It's happened like that. Sometimes in a class you are coming in morning. You are in a mood of, okay, today is the lecture, this madams are there. But after 5 or 10 minutes, topic is started, you are also get gear up. Right? Attention is started to given up. It may happen. It is a phase. Now we are going to learn into it. That is a growth curve. It's called lag phase and log phase. That means lag phase means what? You are in just in a mode of a moving. Yeah, exponential. The log phase will come. That means you are in the state of active condition. Initially, you are coming. Okay, okay. Good morning. Some formalities is going on. Like that also, bacteria will get acclimatized with their own environment right now. So they will able to manage. Okay. Then they will thought, okay, temperature is okay for me. Then the other parameters. If the nutritive material is there, among them, if any hindrance is not there, osmotic pressure is required. Like simple, food is there. We are going to add some salts, some sugars, source material, whatever is there as per their requirement. If out of suppose it's like a 10 to 15 general basic ingredients are there in dishes. Out of this, if the 8 ingredients are not there, suppose you will not like at least one simple salt and sugar, two major things, component. Salt and sugar is not there, you will say, oh, it's food is not good. I don't like it. If it is higher added, salt is added in higher amount. Oh, it's very salty. Yeah. Or it is like a acidic conditions. It's very soft. Someone has added somewhat vinegar more. Okay. What's going to happen? You won't like it. Like that, bacteria will start to ask and see the learn. Okay, what is the environment right now? If it is good, they will start to propagate themselves. It is so. This is the phases are called. Then that part we are going to learn into fourth experimentation. Lag phase, log phase, and the third will come as a stationary phase. That part I'm not looking to discuss right now. But there are the 3 phases are there. That means what? Okay, just simple to example. Like you have started to eat something. It is very good. Your favorite dishes are there. Okay. Maybe for all it is a different. You just eaten up. Oh, it's very tasty. As per mine. Okay, you have sometimes you require some sugar or you have added mixed well. Wow, it's really good. Okay, you have started to eat. Initial phase, it's the leg phase. Then you have started to eat. Initially, it will be a somewhat in a hurry, you like it. 4 foods, 5 spoon, or how you are eating the according to the habits, people are start to eat. For a certain period of time, the tummy start to get filled somewhat. Early it was you are very much hungry. Yeah. Uh, that is the way to start, right? And this is related to growth. Okay, that's why I'm giving the example. But then after what happened? After a period of time, what's happened? That you have taken up, you have eaten up, and your tummy is half filled. Then you are started to talk to one. Your favorite dish is there. Everything is ingredient is going well. But then after what going to happen? You start to halt even. Right? You eating because you liked it. But then the dish is up to a 75% is completed, then you halt. Even it is offered. Sometimes happen that one dish is it's not enough. Okay, you may go again, like suppose the canteen or some area or some friend is there, you are going to offer the second place. But after a certain period, you are going to get saturated. That saturation point is called the stationary phase. That means you are eating it. You are not going to say that no, but you are eating. You will finish the second dish supposed to up to half level. Then you will say, sorry, now it is full. Fully, I'm giving that. I'm giving the everything again free. That is your favorite dish. But afterwards also I'm offering the third dish. You will say no. That means it's a phase called the decline. First phase is the leg. Second lock phase. Third is the stationary phase and the decline phase. That means you start to decline. That now no more. It is like saturations in a class. When you are coming, that's why I'm telling that when you are coming in a class, you are in leg phase. Then half an hour, 15 minutes, you are lock phase. Then you are bearing me, you are in stationary phase. Hour is about to complete. I can understand. You also start, okay, sir, it's the time. Give us some break. So you are start to declining one to another. Happens. So it is the basic physiology of our teaching habitat, teaching methodology. All the things are going in that conditions. All the living system, whether it is related to the learning or it is related to nourishment or any kind of a relationship even. Whatever is there, it is like a parasitic, only kind of a relationships there. There is some kind of a limitations are coming, and they are following this path only. Leg, lock, stationary, and then last is decline. Like it is whenever the people start to interact, for us also, for me and you, first day somewhat basic talk were gone. Second day, you may also able to understand that sir is having some kind of a mentality. I am able to understand the students having some mentalities. Leg to lock phase. After a 1 month, you are coming in stationary phase. Then the decline phase. Symmetry is gone. Sir, please leave us. We will do our own. Okay, so any kind. Okay, and there is some adjustment is required. That is in lock phase. Leg phase is generally a adjustment. Stationary phase is the place. Stationary phase is a place where everything is settled. Some are knowing that sir is going to do this, we have to do. I know the students are going to do this, I have to bear. Both you are going to understand that we have to do this and this. Both the stationary phase is there. But in lock phase is what's going to happen? Full exponential. Because it is like that. It has been instructed to me to do from upper side also. And I'm going to give the instructions to you people, you are going to perform. That is a lock phase is coming. There is no excuses are available. But in stationary phase, because now at the stage that you are start to do that, like attendance, 80% is attendance required. It is 89. I will go in a class. So what's going to happen? Okay, stationary phase, you start to adjust. If the 79% is there or the 75%, oh, sir, I need attendance. Can you manage? Happens. You are in stationary phase. You are asking to manage, and the last end in the decline, whatever 50% is only. So we'll do whatever they want to do. We will do whatever we want to do. Or the 95% is there. Now I want to go in a class. I will manage by myself. That is a self-dependency. So you will decline. So I will take class or not, I will do my job by myself. All the things are working in this physiological conditions. Okay. So let me, what's the time? Wait just a second. Okay, 13 is there, right? Okay, so we will resume again in 8:20. Okay, you may sit here at 8:20 only. I will allow you people to move out. So 8:20 to 8:30, I will give the 10 minutes break. Fine. So about it's 8:15. After 5 minutes, I will give you a break. So we can have just a little talk. So I'm going to end the meeting here for those who are in online mode. Nishit Raichasa. So it means it leg phase, right? Growth is higher and the log. No, in leg phase, there is a growth is very low. But a log phase which will be an exponential phase. Fine, so we will resume at 8:20 again in a meeting. You can take the rest and you can join again back afterwards.